Thank the organizers for the invitation. It's really a great pleasure to be here in Berlin, and I wish us all a very successful meeting. So today I'm going to talk about two different topics. We're going to start with the link between chromatin organization and alternative splicing, and then we are going to talk about evolution of alternative splicing. More precisely, we are going to ask the question, what makes us a unique species? So why alternative splicing became such a big hit following the publication of the human genome? Because we realized that we have 3 billion base pairs in our genome, but only around 21,000 protein coding genes. An average gene is 28,000 nucleotides, composed from 9 exons, separated by 8 introns. Exons, as I'm going to show today, are under selection to remain short, around 130 nucleotides. Introns, on the other hand, show the opposite direction. In lower eukaryotic organisms, they are extremely short, and we have recently demonstrated that they were lengthening during evolution, and if there is something which is the hallmark of the human genome, is that we have some of the longest introns among all living organisms. And the major question that we are going to ask today is how the splicing machinery managed to pinpoint the location of the short exon located between 2 huge intronic oceans. But the big surprise in 2001, when the first draft of the human genome was published, and then after that many other genomes were published, was the finding that total gene number is not indicative of higher level of organism complexity. As for example, a worm, which is basically a tube of 1,000 cells, contains more genes than a fly, although fly is definitely a more phenotypically complex organism. Rice contains twice the amount of genes compared to us, and we have precisely the same amount of genes as any other mammals, including the tiny mouse. So if total gene number is not indicative of higher level of organism complexity, what might be associated with this phenomena? Part of the answer is alternative splicing, in which over the last 5 years, by the use of deep sequencing of the human transcriptome, Ben Blanco, Chris Burge, and others demonstrated that more than 95% of our genes generate more than one type of mRNA molecule called isoform. And from the ENCODE project, we recently learned that we have around 10 isoforms per gene, which therefore enable us to maintain a very small pool of genes and to use alternative splicing as the major mechanism to generate all the transcriptomic diversity, all the different type of mRNA molecules that are needed in order to build and maintain the most sophisticated machinery, namely our human body. And we have demonstrated that exon skipping, this situation, the situation in which an entire exon is included in one mRNA molecule and skipped in the other, connecting the 2 flanking exons together, is the most abundant form of alternative splicing in higher eukaryote, including our body, responsible for approximately 40% of the alternative splicing events in our body. Other forms of alternative splicing are alternative 3' splice site, alternative 5' splice site, and intron retention, the situation in which an intron is spliced out in one mRNA molecule and retained in another one. This is the rarest form of alternative splicing in our body, but this is the most abundant form of alternative splicing in higher eukaryotes, in plants. So we further demonstrated that during evolution, so today when I'm going to talk about alternative splicing, it's going to be mainly exon skipping, the most abundant form of alternative splicing. We have further demonstrated that during evolution, there was a big jump in the level of alternative splicing during the transition from invertebrates and plants to vertebrates, and that we have the highest level of genes that are alternatively spliced. So the good news is that we might not have the highest number of genes among all living organisms, but at least we know how to use them in the most efficient way in order to produce more options via alternative splicing. And in evolution successfulness, everything is about let's try a new options. Species that not try new options usually perished. So higher level of alternative splicing as we advance to human. More than that, we have shown that some of the differences between us, the fact that some of us are tall and short, black eyes versus blue eye, and also some differences in our personality are due to different mode of splicing of some of our exons. For example, some of your exons are constitutively spliced and mine are alternatively spliced. So higher level of alternative splicing as we advance toward human. Exon skipping is the most abundant form of alternative splicing, and some of the differences between us are due to different mode of splicing, especially on exons containing synonymous SNPs, those that do not change the coding amino acid, yet they affect binding sites of splicing regulatory proteins. And currently we are going to get to the main dish. And introns were discovered more than 30 years ago. And we still do not understand how the splicing machinery managed to pinpoint the short islands, which are the exons, located between huge intronic oceans. And this is specifically important in human, in which we have shown that introns were lengthening during evolution. And currently the only way to find gene structure is by sequencing mature mRNA molecules, align them to the genome, finding the clusters of alignments which are exons, and the spaces between them are intronic sequences. So how the splicing machinery is doing it with such high level of accuracy? So our story started when a very talented student of mine, Shragi Schwartz, decided to compile a data set of all human exons that are separated by introns of 1,000 nucleotides or more. And he found a clear signal. Exons contain higher level of guanosines and cytidine, called GC content, compared to the flanking intronic sequences. So what is the meaning of such a signal sitting on the exon? It might be related to the RNA level. For example, binding sites for splicing regulatory proteins. For example, pausing elements during transcription by RNA polymerase II. But it might be related to another level, which is one step above the RNA, and this is the DNA level, more precisely, the way by which 8 histones form a nucleosome. Around it, 146 base pairs of DNA are looping around. And indeed, over the last couple of years, it was shown that a certain fraction of our nucleosomes bind in a sequence-specific manner. And based on these findings, several bioinformatic programs were written that predict where along the DNA a nucleosome going to bind and with what affinity. This is called nucleosome occupancy. One of these programs was written by Eran Segal from the Weizmann Institute, and we used this program across intron, exon, intron structure, and you can see that the program predicted a much higher level of nucleosome occupancy on exons compared to the flanking intronic sequences. So you might say, okay, this is a bioinformatic prediction. We are unsure about the validity of this assay. So we decided to approach a experimental validation. And luckily enough, in 2001, the first whole genome nucleosome occupancy dataset was published. It was done on human T cell, and it was done in order to find the chromatin organization around the promoter region, leaving us a lot of breed inside gene body itself. And as you can see, we detected a much higher level of nucleosome occupancy on exons compared to the flanking intronic sequences. So exons are already being marked in the DNA level, a mononucleosome marking the location of exons compared to the flanking intronic sequences. Is this something which is unique only to us? The answer is no. This is a highly conserved process going all the way from human till worms. So why a mononucleosome sitting on exon? Probably it's marked for some important function. What this function is. On the right panel, you can see the average length of internal exons, exons which are flanked by 2 introns. And you can see that in all of these organisms, there is a very sharp peak, indicating that exons are under selection, not to be too short and not to be too long. The height of this peak is identical in all different organisms, around 127 nucleotides. So why exons are under selection? To maintain a fixed size. The green line indicating the length of DNA that is protected by a mononucleosome, which is 146 nucleotides. As a matter of fact, the core nucleosomes protect 127 nucleotides, which is precisely the length under which exons selected for. So if this is a coincidence, I don't think so. I think that chromatin organizations somehow tell the splicing machinery, here the exons are. And indeed, we have shown that changes in chromatin organization can affect alternative splicing. More than that, if we are tackling directly the splicing machinery, we've demonstrated that changes in splicing by itself can affect chromatin organization. So how these 2 layers talk with one another? They seem to be a totally disconnected layers, DNA level, chromatin level, RNA processing level, how they communicated. Most probably via RNA polymerase II. It was shown that elongation rate of Pol2 can affect alternative splicing. Pol2 by itself holding splicing factors attached to the CTD domain, and more recently, specific histone modifications were shown to hold splicing factors on them, especially H3K36 methylated 3, which is enriched on exons, on nucleosomes located on exons. So all of these results together suggest that there is some kind of a crosstalk between RNA polymerase II when bumping into a nucleosome located on exon, and the emerging of RNA, which is telling the splicing machinery, this is the RNA that you need to process, and putting splicing factors on this exon. So it's ignite the exon selection process. So we learned from these results that nucleotide composition, especially across exon introns, are very important characteristics of gene structure. So what do we know about nucleotide composition all across the human genome? Are the 4 nucleotides distributed equally all over it? The answer is no. Bernardi have shown many years ago that there are regions along the human genome, megabases along these regions, shown here by the hot colors, containing many genes within them, along with intergenic sequences between one gene to the other, with very high GC content region. This is, for example, a situation for chromosome 1. And they are located alongside with regions shown here in blue megabases long, many genes inside them, with very low GC content regions. And in order to avoid questions at the end, 70% of our genes located in the low GC content regions and 30% located in the high GC content regions. And there is no gene ontology, there is no specific function for genes that are located in the high or in the low GC content regions. The high GC content regions are newcomers into the genomes. They are restricted only to warm-blooded organisms, meaning to us, to mammals, and avians. Outside, in cold-blooded organisms, there is one structure, which is low GC content regions. We don't know the reason why certain blocks along genomes became heavily mutated toward high GC content region. There are some models. One of them is called bendability, and bendability is basically chromatin organization. You know, from the gizma staining, you can see different bands, which might be related to high and low GC content regions. There is a later model suggesting that this is relating to timing of DNA replication, but there is still a lot of room for research in that field. Nevertheless, we decided to ask what is the situation in terms of gene structure inside genes that are located in high and low GC content regions. And to make a very long story short, we found two different gene structures. Genes that are located in low GC content regions contain long flanking intronic sequences with low GC content. Crossing the bridge between the intron and the exon, you can see elevation in the level of GC. We call it a bump. And the differential GC content at the 3' splice side, at that side of the intron, is much higher than on the other side of the intron-exon junction. This is the 5' splice side. And this is due to a gradient of GC from one side of the intron to the other side of the intron. So genes that are located in low GC content regions have long flanking intronic sequences and a differential GC content between the intron and the exon. What about the situation of genes that are located in the high GC content regions? They have short flanking intronic sequences with very high GC content regions, and there is no differential GC content across the intron-exon junction. And yes, we noticed that here in the first 50 nucleotide in the intron, we have a bump, higher GC content. We believe that this is a splicing regulatory sequence, and we are currently working on it. So we have two different gene structure for genes that are located in low and high GC content, one with a long flanking intronic sequences and a differential GC content, the other one short flanking intronic sequences and no differential GC content. This is again something which is unique to us. Of course, the answer is no. This is related, the separation into two different gene structure is related only to warm-blooded organisms. Outside of this clade, this is one situation, short flanking intronic sequences and a differential GC content between the intron and the exons. And in collaboration with Tal Pupko from Tel Aviv University, we reconstructed the ancestral stage, which is precisely like that. So during evolution, with the transition from cold to warm-blooded organisms, genes that remain in the low GC content regions, their introns were allowed to lengthening. And as long as they have the differential GC content between the intron and the exon, their splicing is safe. Genes that became to be involved in regions that shown elevation in the GC content regions eliminated the differential GC content between the intron and the exon. However, they are under selection to remain short. The separation into these two structures of gene structures suggested to us that we might find the old hypothesis that suggested more than 20 years ago that there are two different mechanisms for intron-exon selection, which are called intron definition and exon definition. According to this model suggested by Suberjet more than 20 years ago, introns that are selected via intron definition, whenever they have mutations, it's going to lead to intron retention. On the other hand, exons that are selected via exon definition, mutations in those genes, going to lead to exon skipping. Can we prove that indeed these two gene structures are related to intron definition and exon definition? In order to address this issue, we compile a data set of disease-associated mutations leading to exon skipping, more than 360 cases, and we measure the GC content across the intron and the exons. And you can see the differential GC content, which is similar to genes that are located in low GC content regions. So genes that are located in low GC content regions are associated with exon skipping. You mutate the gene, it's going to lead to skipping above this exon. What about mutations that are going to lead to intron retention? So we decided to compile a similar data set, and remarkably, except of two recent publications, there are no indications that mutations that are related to diseases cause intron retention. We don't really believe that there are no diseases like that, but we believe that whenever you have intron retention, the intron-containing RNA remain in the nucleus and sent for degradation, and therefore you cannot sequence for those patients any type of RNA related to the disease. So in order to overcome this problem, we compile a data set of 3,000 cases of tissue-specific intron retention events, and we measure the GC content across the intron-exon structure, and the GC content is surely enough high as the level of genes located in the high GC content region, and the differential GC content between the intron and the exon is minimal. So we believe that we identified two different gene structures that are related to two different mechanisms by which the splicing machinery approaches introns or exons. And if you're going to give us any kind of gene, we are going to predict a priori what is going to be the outcome of splice site mutations. We further demonstrated that as long as you have the differential GC content between the intron and the exon, splicing is safe. You can lengthening the flanking intronic sequence or shorten them to less than 100 nucleotides without affecting the splicing machinery. We further demonstrated that the differential GC content by itself can regulate alternative splicing. The higher it is, more exon inclusion, better detection of the alternative exon. The lower the differential GC content, more exon skipping, less detection of the exon during alternative splicing. And we experimentally validated these results. What about chromatin organization? Only when you have the differential GC content between the intron and the exon, there is a mononucleosome marking the location of the exon already in the DNA level. However, genes that are located in the high GC content regions have many nucleosomes located all over gene structures, but there is no mononucleosome marking the location of this gene better than any other location along these genes. And this suggested to us that it might be that there are other markers sitting already in the DNA level, telling gene structure in terms of exon-intron structure. And what additional markers might be? Of course, epigenetic marker. And the most important epigenetic marker that is related to nucleotide composition is DNA methylation. When we have the dinucleotide C following by G, there are certain enzymes approaching the C and adding a methyl group on it. This is called CpG methylation. We know a lot about CpG, about the function of CpG methylation in the promoter region. This is the work of Martin Winograd. But we don't know much about the structure of DNA methylation inside gene body. Luckily enough, in that time, the first whole genome DNA methylation was published. And we use this data set, which is from human embryonic cells, but we did it also for many other cells and tissues, and the results are pretty much the same. And we examined DNA methylation across low GC content regions, which is in green here, compared to high GC content regions, which shown here in blue. In the low GC content region, 85% of the CG site in the intron sequences are methylated. Crossing the bridge between the intron and the exon, you can see elevation of 5% in the level of methylation. So in this gene, there is a mononucleosome marking the location of these exons. And in addition, there is a higher level of DNA methylation within those exons. What about genes that are located in the high GC content regions? 65% of their CG site are methylated inside the introns. Getting close to the intron junctions, there is a drop of 10%. Crossing the bridge between the intron and the exon, elevation of 17%. So the differential GC content here is much higher than in the other group, suggesting that although that in this gene structure, nucleosomes do not differentiate between exons and introns, at least at the DNA level, DNA methylation can differentiate between intronic and exonic sequences. We further demonstrated that genes that are located in high GC content regions, whenever they have CG at specific positions at the 3' splice site or 2 positions at the 5' splice site, whenever those sites are C and G, they are 100% methylated, suggested that somehow DNA methylation tells the splicing machinery for those exons, here is the right splice site. So we have shown 2 different gene structure for genes that are located in high and low GC content region, 2 different chromatin organization for those 2 families, and 2 different mode of epigenetic markers, which are CpG methylation, DNA methylation. We are currently going to shift gear, and for the last 7 minutes, we are going to talk about evolution of alternative splicing, more precisely our own unique evolution. We're going to ask the question, what makes us a unique species? In 2002, we identified a novel evolutionary mechanism creating primate-specific genomic diversity, namely mRNA molecules that are unique to our species. We call this process exonization, and exonization is a birthing process of new exons from what used to be intronic sequences, meaning introns became to be exons during evolution. Remarkably, in human, the majority of the new exons originated from a primate-specific retroelement called Alu, or Alu. Alus are 300 nucleotides long. They were so successful in spreading in the human genome so that currently we have 1.1 million copies of Alus located all over the human genome, and we found that more than 700,000 of them were inserted into intronic sequences. Some of them that were inserted into intronic sequences gained the right mutations. As a matter of fact, they have those sites already prepared for them. So these mutations activated 2 splice sites, 3' and 5' splice site, so that the splicing machinery can start recognizing a certain region of them as a new exon, an Alu exon. Our results indicate that these exons are only alternatively spliced, meaning they enrich our human transcriptome with new options containing Alu exons without eliminating the old options that needed in order to synthesize a functional protein. Whenever the mutations creating a strong exon that is inserted into all mature mRNA molecules that are generated from this gene and eliminate the old mRNA molecule, it inflicts diseases like Alport and Slay syndrome and OAT deficiency, meaning 3 different genes, 3 different Alu elements, the same molecular mechanism behind those diseases. And this is not a dismissible process. We have more than 1,060 alternatively spliced Alu exons in our genome. And we have demonstrated that some of these exons already gain functions that are unique to primates, functions that might be responsible for some of our unique characteristics as primates and maybe even human. Most of the Alu exons are currently non-functional. We can live with a certain noise in the system. But they are our future evolution. If more mutations occurred and the right open reading frame will be open in those exons, it will elevate or advance our speciation. And unfortunately, there is a cost. And the cost is that a certain fraction of those exons are associated with cancer. So on one hand, this process advances our species, and on the other hand, it inflicts diseases, genetic disorders, and cancer. So exonization, and especially exonization of Alu elements, create new alternatively spliced exons. In 2007, we identified a second mechanism for the origin of alternative exons, and those are exons that shifted their mode of splicing from constitutive to alternative splicing. Let's take, for example, this gene. In zebrafish and chicken, the exon is constitutively spliced, and in all mammal, it is alternatively spliced. And we found the molecular mechanism for most of those transitions, and those are point mutations that were inserted in the splice signals, weakening their location, implying that in one mRNA molecule, the splicing machinery managed to find those splice sites and splice the exon in. In another mRNA molecule, the splicing machinery missing this signal, skipping the entire exon. And we have shown that this is not restricted only to exon skipping, because also alternative 5' and 3' splice site originated from the same mechanism, by which point mutations, either within an existing exon or in one of the flanking introns, creating a new splice site that start to compete with the old one. And we have shown that under these conditions, the new alternatively spliced region is under selection, not to change the open reading frame for the rest of the mRNA molecule and not to insert a premature stop codon, and we have validated it experimentally. So based on all of these results, we can conclude that our human genomic complexity is an outcome of ongoing processes in which new options are tested. Some enhance biological system and thus leading to higher level of complexity and speciation. Others are deleterious and thus causing genetic disorder and cancer. Unfortunately, I see that I spoke too fast and I'm ahead of time, so I'm going to slow down for the last part of my talk, which is mostly summary. Okay, so we've shown today that exon skipping is the most abundant form of alternative splicing in higher eukaryotic organisms. We've also demonstrated that during evolution there was elevation in the level of alternative splicing and that we have the highest level of genes that are alternatively spliced. We've also talked about the fact that some of the differences between us are due to different mode of splicing of some of our exons, constitutive and alternative spliced. We found that the ancestral gene structure contains short flanking intronic sequences with low GC content. Crossing the bridge between the intron and the exon, you can see a bump, higher level of GC content inside the exons. During the transition from cold to warm-blooded organisms, two structures appeared, genes that remain in the low GC content regions. Their introns were allowed to lengthening, for example, by insertion of Alu elements. And as long as they have the differential GC content between the intron and the exon, their splicing is safe. Genes that became to be involved in regions that became with high GC content regions elevate the GC content in the intron and the exon, and the differential GC content was eliminated. But their introns are under selection to remain short. For this group of gene structure, mutations are leading to intron retention. For genes that are located in the low GC content regions, they are associated with exon definition, meaning if there is a problem to identify the exon, there is exon skipping leading to ligation of the two flanking exons. We have shown that the differential GC content by itself can regulate alternative splicing. The higher it is, more inclusion in of the alternative exon. The lower it is, more skipping over this exon. Only when we have the differential GC content, a mononucleosome marking the location of the exon already in the DNA level. For genes that are located in the high GC content region, there is no chromatin organization that mark their location, but they have a different epigenetic marker, and this is DNA methylation that differentiate between intronic and exonic site, which is also applicable for genes that are located in low GC content regions. And the linker between these two seemingly two different worlds is presumably RNA polymerase II. RNA polymerase II, when traveling along the gene, it carries splicing factors, hitting a nucleosome located on exon, well-defined nucleosome, probably slowing down. There are certain indications that indeed it's need to slow down. Slowing down of POL2 was shown by Alberto Kornblit from Argentina to change the splicing activity for some exons. And there are certain splicing factors already attached to this nucleosome to start with, attached to chromatin organization, suggesting that when the 3' splice site emerge first and then the 5' splice site emerge after that, splicing factors are moving from chromatin and from RNA polymerase II into the right side, igniting the exon selection process, presumably explaining why the length of the flanking intronic sequences is not important anymore, because all the signals are currently located on the exon itself. So who are the people that were involved on all of this project? I cannot give credits to all of them, but for some, yes. The first part of my talk regarding higher level of alternative splicing and differences of alternative splicing between different individuals was done by Ido Kim, currently with Google, and Amir Goren, bioinformatically, and experimentally it was done by Mariana Amit. Shragi Schwartz found that nucleosomes mark the location of exons already in the DNA level. He's currently with Aviv Regev and at the Broad Institute. You're probably going to hear a lot about him in the future, a very talented person. The GC story was done bioinformatically by Ido Kim, Amir Goren, Dror Hollander, and Sar Galfman, and experimentally by Mayan Amit, Maya Donio, and Galit Lev Maor, my longtime lab manager. The last part of my talk regarding the exonization of Alu elements was identified by Rotem Sorek, who is currently a PI, a group leader at the Weizmann Institute, and experimentally by many people, mostly by Galit Lev Maor. I would like to thank you all for listening, and I will be happy to take questions. Thank you very much.
Now, this is the usual problem. Thank you. Questions. Now, of course, you understand that I can't see very well. Okay, I can see Alfonso. You move your hands because it's difficult to see from here. Please, Alfonso.
Thank you for the talk. Impressive work, impressive talk.
Thanks.
You mentioned at the beginning the role of timing of replication. I wonder what you think about how to put timing of replication into the model.
Not my field. You will have to ask people that suggested this model. I have no idea. Probably they are right, because in order to open the DNA to put DNA polymerase inside it, the melting point is very important. So they might be right, but I can't elevate any more about it.
There is probably another question over there. Yeah.
So have you looked to see whether genes move between high GC and low GC?
I'll repeat the question for the recording.
I'm going to repeat the question,
Yeah,
whether genes move from high GC to low GC. Long story short, no.
Question? Yeah, thank you, Gil. I have a question concerning the Alu, the exonization. Can you comment on the Alu and their relation to nucleosome? Because somehow you skipped this and you mentioned it in the second part of your talk. Thank you.
Good question. This is something that we are currently doing in the lab. There are two arms in the Alu element that originated from duplication. This is basically 180 nucleotide duplicated twice with a spacer of poly T located between them, and poly T reject nucleosome occupancy. There are many indications that there are two nucleosomes sitting on the right hand, on the right arm and on the left arm. There is a beautiful story about DNA methylation and how DNA methylation blocked the promoter located within the Alu element. We are currently testing correlation and validation between exonization and high level of nucleosome occupancy in exons that tend to exonize, but don't tell it to anyone else, please.
Only 1600.
Okay.
Yes, the question is, first, thank you for the talk. And the question is, do you have any data on population dynamics of alternative transcripts?
Say
in the time frame of compatible with, say, well, energetic cycle,
you will have to start the question from the beginning. I have a problem hearing you.
Do you have any data on population dynamics of alternative transcripts in certain time frames, say compatible with energy oxidative phosphorylation cycle or circadian cycle, or anywhere, any other time frame?
Okay. In terms of evolution, we have shown that the majority of genes that change their mode of splicing from constitutive to alternative splicing, it was during the transition to mammalian organisms. So in mammals, we see a very high jump in the level of alternative splicing. Any other questions related to that, I have no idea.
So within 2-3 hours of each other, the relative quantities, abundances of transcripts.
Okay.
Do they
What you are
change?
asking,
How do they change?
Okay. What you are asking is how fast exons change their mode of splicing from mostly exon inclusion to mostly exon skipping. Am I right?
Yes, that would be correct.
Okay. So yes, there is work. There are certain works, especially from the lab of Ben Blanco, measuring the timing of changes in alternative splicing. The most interesting scenario is in sex selection in Drosophila, in which a gene called sex-little changed the mode of splicing very rapidly within less than half an hour.
I'm curious about polarity effects or location effects of these, for instance, the exon islands. If you noticed in any of your work that where in the transcription unit this exon is, you see variation or signal.
Okay, so you basically ask whether the exonization of Alu elements have a very specific location within intronic sequences.
Or for that matter, the other parts of your talk, other aspects of the biology you've talked about depend in any way on the location within the gene.
Okay. So we have shown that the longest introns are located close to the 5' end of gene structure. No, the first intron is the longest one, and then they are starting to be shorter and shorter. Exons that tend to be exonized in the beginning of the gene, and if they are functional, they are usually divided by 3, meaning they are not going to change the open reading frame. You are getting more close to the 3' end of the gene. There are less exons that when they are produced anew, they tend to be divided by 3, meaning you can have the C-terminal domain of the protein with some sort of flexibility. But no flexibility is allowed in the beginning close to the N-terminal. This is the best that we know.
Gil, nice talk, very nice talk. A lot of what you've shown, these are summaries over a large number of exons. Can you say something about the appearance of these signals, such as the jump in GC content or the exon length at the individual exon level? For example, could you train a classifier to identify exons better this way?
I have problem hearing you here, Ken. So can you repeat the question?
Yeah. A lot of what you've shown in terms of the jump in GC content or in the tendency for exon lengths these are summaries over a large number of exons, but the spliceosome has to identify individual exons.
Yeah.
Can you say something about what these signals look like at the individual level?
Okay. So Chris Burge and also my lab demonstrated that approximately 20% of the splice signal arrive from the splice site itself, the 5' and the 3' splice site, the signal that is now the polypyrimidine track and the brain site sequence. On top of it, we don't know precisely the fraction, but the fraction is most probably around 40% from the sequence within the exon itself. And those are binding sites for splicing regulatory proteins that are known mostly as SR proteins, and they love to bind GC-rich sequences. And the most important binding sites for those SR proteins are located in the first 20% and the last 20% of the exon sequences. Over the last several years, it was shown that in the beginning of the introns, there are also many signals. For example, signals that love to bind, the protein that love to bind AU-rich sequences such as TIA1 and TIAR and PTB. And they tend to regulate, and they belong to a different class of protein that are called AGNRMP proteins. And they bind mostly to intronic sequences, but they need to be close to the exon. But I'm quite sure that this is due to technicality in experimental validation. There are most probably other sequences located way outside it. We have also shown that secondary structure play a very important role. If you have secondary structure that is located before the exon, it can block the exon selection process. After the exon, in the downstream intron, it's reduced the problematic. So there are many correlations that need to be taken into consideration, and there is several beautiful papers, mostly from the lab of Ben Blanco, that summarize all of them in what is known as the splicing code, taking into account all of them, telling you a priori how your exon is going to be regulated under these conditions. And there is a lot of room in this field.
Are the splicing signals that you're talking about for your exons, are they maintained in pseudogenes, non-processed pseudogenes?
Oh, the answer, long story short, no. All of these publications were done with a data set of reliable pseudogene, no signals is found.
Is there a role for histone modifications? Maybe it be repressing or activating exons?
Can you start over?
I'm asking whether there's a role for histone modifications in alternative splicing.
Yeah.
Do you know anything about that?
Okay.
Is anything known?
The most known histone modification, which is located on exon, is H3K36 methylated 3. That was shown to be associated with a protein that binds a splicing factor, which is called PTB. And it was shown that if this binding is happening, that this binding is most probably triggered by the splicing machinery, that this modification is being triggered by the splicing machinery, more splicing, more histone modification on these exons, and those exons are enriched with this modification. There are two additional histone modifications, I think H3K79, that were also being shown to be enriched on exon, but currently their function is largely unknown.
You mentioned the exonization by Alu element.
Yeah.
Did you also work on like other repetitive elements like L1 in the human exonization of those? And how often are these different situations?
We published a paper in which we have examined exonization of all repetitive elements, including L1. L1, which is an older retro element, tend to exonize less. And the most important candidate to examine, compare exonization, is the B1 in mouse. And the reason for that is that B1 is the monomeric form of Alu, whereas Alu is the dimeric form. The major reason that retroelements tend to exonize is that they have a poly A tail in their 3' end. But since they can insert it into introns in sense or antisense orientation, whenever they are in the antisense orientation, this poly A is currently poly T. And poly T, we have shown, is the most important signal for the exonization process. This is the polypyrimidine track in the 3' splice site. And from there, this is a piece of cake to start the exonization process, just to search for AG sequence located downstream of it, reaching ahead for 127 nucleotide to search for GU as the 5' splice site. You're going to find something over there, and that's what ignites the exonization process. So B1, much less exonization. Alu with a lot of exonization, because these two polypyrimidine tracks mark the location of exon from both sides. So Alu is very unique.
So for what you have said, it's not clear to me how, are you hearing properly? Okay.
Yeah.
For what you have said, I don't understand how alternative splicing can be regulated, because it looks pretty much as a deterministic process, right? I mean, you have the signal or you don't, so you recognize the exon, and maybe there is some probability that it will be taken or skipped, but how do you regulate that in order to provide a different isoform?
Okay. You want to stab me in the back. Okay. So the spliceosome is the biggest complex inside our cell. We have more than 100 proteins that just play regulatory function. They are expressed differently, for example, between neuronal cells and somatic cells. And they are also being regulated by phosphorylation. So some of them need phosphorylation in order to activate. So this is a very complex mechanism in order to regulate alternative splicing. Forget about what you've said. This is the most regulated system in our body.
I also have a question. I was waiting for the audience to calm down. So you sort of hinted at that, but didn't get into details. This exonization of Alu bring about new isoforms. What about their function with respect to the non-Alu, non-exonized ones? Do we have any example which is...
Okay, so what Anna is basically asking, what is the function of the 1,060 new Alu exons? Okay. So for 61 of them, they produce the same open reading frame leading to phosphorylation of certain protein. There are certain classes, less than that, 5 here, 10 here, that produce the same function. But each Alu reached to its exonization via a totally different story. Deletion here, insertion there, and therefore the open reading frame, although that most of them use the same site as the 3' splice site and another site as the 5' splice site, the open reading frame, most of them do not have a real open reading frame, but those that have open reading frame have a totally different open reading frame due to the mutations that led to their exonization. Okay, thank you all.
So on behalf of everybody here who enjoyed your talk so very much, thank you.
Thank you.
Thank you.